everyone, welcome to this course Fundamentals of Computational Biology. This is lecture 1 and I will be presenting an overview of the course. Before we dive into the topics we will study, let me introduce our team. We have Professor Tanushree Chatterjee as course coordinator and I will be your course co-coordinator. Additionally, we have Dr. Sharmistha Banerjee, Dr. Kush Nayak, Mr. Abhinav Jagtap, Dr. Santi Swarup Dubey, Dr. Animesh Sharma and Mrs. Silpi Banerjee as experts. Allow me to provide a brief introduction to this course, Fundamentals of Computational Biology. This is an interdisciplinary approach to study the complex interactions within the biological systems. Through experimental techniques and mathematical modeling, we aim to understand the underlying mechanism that governs the cell and entire organism. This course spans 12 weeks and consists of 40 different modules. In the first week, we will cover the fundamental principle and mathematical concepts. In the second week, we will be focusing on the model building part. In the third week, we will apply these models to basic concepts such as bacterial growth kinetics and enzyme kinetics. In the week 4, we will involve into the learning of population dynamics and data representation. In the week 5, we will be dedicated to solve the mathematical modeling aspects. Week 5 and 6 will be dedicated to solve mathematical models. In the week 7, we will explore the intriguing phenomena that exist in biology such as game theories. Week 8, we will delve into the various human systems such as respiratory systems, circulatory systems, etc. In the week 9, we will tackle more complex processes such as transcription, translation, mutation, evolutionary processes, etc. In week 10, we will be learning these mathematical modeling that specifically targets evolutionary processes. In the week 11, we will explore the highly useful phenomena such as flux balance analysis and metabolic switches. The last week will be the revision of data representation, the mathematical modeling and program building part. I will now discuss examples where mathematical modeling will be useful. First, let me talk about biological models and biological interactions. Before that, let's understand what is a network. In a simple sense, a network interaction is the interconnection between multiple bodies. For example, I have studied BTech program where I might have connections with my batchmates. These batchmates after completion of the degree move to different locations and develop connection there. This can go to several different levels. I can preassumbly say that in my network at least I have interacted with maybe 10,000 or maybe 50,000 people. Now, keeping this in mind, let's understand the biological networks. Biological networks can be incredibly complex with different components continuously interacting with each other. These components include genes, proteins and other molecules, all playing important role in maintaining the checks and balance of a cell. One way to understand this interaction is through biological network analysis, which visually represent how different components are connected. Mapping these connections helps gain valuable insight into biological system and how changes in one of the components can alter the dynamics of the entire functioning of a cell. Biological networks are powerful tool to understand the complex interaction in living system. By visualizing these interactions, the researchers can develop insights into the fundamental life processes, enabling development of new therapies, new treatment processes that can improve the human health. Various methods including clustering, centrality analysis and motif analysis can be used to study biological networks. These techniques helps identify the structure in a larger data sets, enhancing our understanding of human biology. Not only the human biology, but these are incredibly used to study the bacterial network, the virological network, as well as in a more complex form a socio-economic status can also be studied with these network analysis. Now, the emerging research is driving innovations in network science with a focus on integrating data from different sources and studying networks across multiple scales, such as the whole organism or the entire ecosystem. A key challenge in this area is developing methods to analyze, visualize these complex networks, which may involve millions of different components interacting in a specific way. For example, in a normal disease pathway, what happened is a particular organism or a microorganism infect a host, let's say human beings. These hosts, after entering to a system, interact with various cellular proteins, and some of them might interact with host DNA. So, in this way we can analyze the various network by which the foreign component, that is a bacterial component and a host component, how these two are interacting with each other. Next, let me explain the cellular signaling pathways. These pathways involve cell to cell communications, where a single cell communicates through signal and activates protein in response to different protein that are part of the host cell. These are also affected by environmental signals. Cell to cell communication happens via membrane receptors. Many pathways rely on cell membrane receptors that binds to specific ligands, transmitting signal across the membrane and finally reaching to the cell. Signaling pathways can also activate transcriptional factors, regulating gene expression and leading to changes in a cellular behavior. Now, let's talk about the use of differential equations in the modern building part. The mathematical model simulates the behavior of biological system over time, allowing us to investigate the complex dynamic phenomena such as oscillations and feedback loops. These are the complex processes which we will study in subsequent classes, but for today's lecture, let me just talk about a brief introduction part, how these aspects will be useful in the future studies. Now, differential equations are commonly used for their abilities to capture the time dependent behavior of these biological phenomena. The kinetic parameters govern these differential equations must be estimated from the experimental data involving methods such as optimization and Bayesian interference. We will study all these phenomena in subsequent classes, so don't worry about today's lecture. Next, I will be discussing about reverse engineering biological networks. Reverse engineering is a powerful technique used by scientists to gain deeper understanding of biological systems. This approach involves using of experimental data such as gene expression or protein-protein interactions to construct complex network that represent the underlying structure and function of biological system. One of the major advantages of reverse engineering is that it allows researchers to explore the complex interactions that occur within the system or the outside of a system. By analyzing these networks, scientists can gain new insights into the mechanism that governs biological processes and develop more accurate and structured model for these systems. However, the reverse engineering is not without its challenges. Constructing accurate network from the experimental data requires sophisticated data analysis technique, as well as the deep understanding of these biological phenomena. These systems, additionally, the data used for construction of these networks is oftenly noisy and incomplete, which makes the processes of reverse engineering even more difficult. Let's talk about a common phenomena here. For example, in healthcare industry, we will be continuously needing certain amount of supplements such as vitamins. Now, we all know in a normal processes these vitamin synthesis might take forever to reach a industrial scale production, what we do is we understand these biological networks, how each components are interacting with each other. Now we take the help of reverse engineering, we increase the concentration of our desired enzymes or these vitamins by altering the speed of these synthesis processes. Despite these challenges, reverse engineering continues to be a valuable tool for scientists in a wide range of fields, from biotechnology and medicine to environmental science and beyond. By leveraging the power of these techniques, scientists are able to gain a new insights into the complex system in the underlying world and develops solutions to some of the most pressing changes facing society today. I will end today's lecture by speaking about metabolic network representation. Now, metabolic network, as we understand, these involves the metabolic component of a single cell. For example, a metabolic network can include the expression of proteins as well as the transcription factors that involve into activation of these genes. Now, in a metabolic network there can be several components. Now if we increase the complexity of a metabolic network, we can see a lot of different metabolic networks can interact with each other. For example, let's take a simple example of glycolysis. We all know what is a glycolysis. In a glycolysis pathway, glucose enter to a cell and after a series of alteration it is converted to pyruvate. We know these processes. Now there are several enzymes that are part of these processes. These enzymes convert one component to another component and finally we will reach to a final product, that is pyruvate in this case. Now pyruvate is incorporated into a different cellular pathway that is called as TCA cycle. So now this TCA cycle as well as the pyruvate cycle both interact with each other. So if I talk about metabolic network, now this includes one metabolic pathway or several different metabolic pathways. When I speak about metabolic pathway, these are series of chemical reactions that convert one molecule to another molecule. Now in a complex metabolic pathway, the end product or the desired product production can be altered with the help of the change in speed or the concentration of each of these enzymes. Now since we have to increase the concentration of a desired product, we should have a thorough knowledge of these metabolic networks. These metabolic networks ranges from bacteria to viruses to human beings. Now there are several different databases that studies this metabolic pathway and have a detailed representation of this metabolic pathway, such as KEGG pathway or we all know KEGG databases. These KEGG databases have categorization of these metabolic networks or metabolic pathways. We can take example of one metabolic pathway and we can study if the similar pathway exists in a one organism or another organism. Now we all are biologists, we have been studying the concept of cloning and etc. in a different level, different understanding of scales. Take example, in a cloning process you will be requiring the knowledge of each of these proteins or each of the genetic components involved in these pathways. Now if I talk about metabolic pathway and doesn't talk about its genetic expression, it will be a kind of incomplete. So in a subsequent lecture we will be studying these individual processes. The aim of this course is not to discuss a lot of biological pathways or lot of biological processes, but to teach you guys how we can take the knowledge of existing biology and we can convert these existing biology to a more understandable approach. That is the aim of this course. For example, I will talk about a very brief idea how we will be taking these biological processes and converting to a newer form of science. Just like you, I was also a student of biology. I was afraid of mathematics. So Now since I have done the research in this particular area, I know what is the importance of mathematical modeling and a complex computer science knowledge that will help in understanding of a lot of biological processes in a newer way. Let's take an example of the recent COVID case. So we all know the COVID viruses entered to a human system and multiplicate itself. Now these viruses spreads from one human being to another human being with the help of some medium. That medium can be touch, that medium can be direct in a very close contact with each other. How this course will teach you is that the mathematical reasoning behind these spread or these rapid spread. Now one individual is infected with coronaviruses. These individual can now spread the viruses in its vicinity. Any persons which comes in a close contact with these vicinity can take up these viruses from infected individuals, and now that person becomes carrier. Now how we will represent this mathematically is, let us consider the n concentration of these viruses are required to infect a healthy individual. Now after infecting with these individuals, one healthy individual that turns to a susceptible and finally to a diseased one can produce copies of these n times viruses. Now these n viruses can infect m number of individuals. That m number of individuals can infect many multiple individuals. So in a mathematical sense, these follows exponential growth. We all know what is the formula of exponential growth, that is e to the power x. And if I want to say that these have certain limits, when it reaches to a certain limit, it will follow a complex saturation curve, that with the help of figure we can understand what is a saturation curve. That means at the end it will reach to a saturation level. When it reaches the saturation level, we all know what is the mathematical function behind it, that is a times x to the power n divided by a into x to the power n plus a constant, that is k, that represent how rapid is the processes. Now we know that what are biological networks and metabolic networks. Let's discuss another important phenomena, that is kinetic models and dynamic simulations. Kinetic models and simulations are like powerful tools that scientists use to study how living things work. Imagine it is a computer game where scientists create a virtual world of biology. They can now see these tiny parts inside our body, how they move and interact over time. These simulations help scientists to learn many things. They can explore the processes that are difficult or unsafe to study directly. It's like a safe virtual lab where they can experiment without any risk. By understanding these smaller parts, they also figure out how everything works together on a bigger picture. One cool thing is that scientists can predict what happens when they change something in the simulation. For example, they can now test how for a new medicine might affect our bodies before giving it to real people. This saves times and helps them for the better decision making capacity. When scientists combine these simulation with another techniques like genetic and imaging, they can get a clear view about the biology. It's like a putting up a puzzle pieces together to see the full picture. This way they can learn new things about living systems and make exciting discoveries that can help improve our healthcare and well-being. So these kinetics models and simulation are like super tools that bring up closer to understanding the amazing world of biology. We will discuss some application part of what we have studied in today's lecture. One important aspect is enzyme kinetics. We all know what enzymes are and what are the function of these enzymes. Enzymes play a crucial role in our body, helping with important chemical reaction that keep us alive. To understand how these reaction works, scientists use spatial models called as kinetic modeling. These models are like tools This shows how enzymes and their ingredients called substrate interact and how external factors like pH and temperatures affect them. A very famous equation known as Michaelis mental equation helps us understand how fast these reactions really happen. It tells us about amount of substrate affect the reaction speed, etc. Scientists also look at how pH levels and temperature impact these enzymes. Just like how too much of heat can make us feel unwell, extreme temperature can affect enzyme too. Each enzyme has an ideal temperature where it works at its best capacity. Additionally, advanced models consider their factors that influence enzyme activity. Think of it as a different switches that can turn enzyme off or enzyme on, controlling their behavior. The cool thing is that these models have practical uses too. For example, they help in developing new medicines to target specific enzymes that treat disease without harming other parts of the body. For example, in today's world we are using a lot of medicines or antibiotic that targets a specific enzyme in these organisms. Now these enzymes either lose their ability to perform any reaction at all, or they lower down their speed of reactions. Anyway, they inhibit these microorganisms to further grow inside your body. So by using these kinetic models, scientists unlock the secret of enzyme reactions and find ways to improve our health and well-being. Another example is study of oscillations and feedback loops. Dynamic simulations are like a helpful game for these scientists. They use this simulation to understand how living things like plants, animal, microbes, human beings work inside their bodies. It's like peeking through a tiny world where all these tiny parts interact and create a different patterns and rhythms. One exciting thing dynamic simulation teaches is about our internal clock called as circadian rhythms. Have you ever wondered why you feel awake during the day and sleep at night? Well, this simulation helps scientists to figure out. They show us how special genes and proteins inside us work together in a feedback loop, just like a dance. This dance keeps our bodies sync with our 24-hour day schedule, about day, about night. One another cool thing is you must have seen some of the flowers which only blooms at particular season, and during the night they cannot bloom at all. So these help understanding how these decision making process are taking up inside a system, or you can say a whole organism. Another cool thing these simulations reveal is how cells changes and become a unique type with specific jobs. This process is called as cell differentiation. Imagine your body as a big team and each cell is a player with a specific role. Dynamic simulation allows scientists to play with these teams, changing some players to see what happened next. It's like experimenting with a puzzle to understand how pieces live together to make a whole picture. By using these simulations, scientists can also predict what might happen if certain changes happen inside our bodies. For example, if there is a tiny mistake in our genes, it can lead to a health issue. Take example of sickle cell anemia. A tiny change in just one locus inside the entire DNA can make a person sickle cell affected, and they cannot perform as equal to a healthy individuals. With dynamic simulation, scientists can see how these mistakes affect the team and come up with an ideal solution. Overall, these simulations are essential tools that help scientists to learn more about us and how to keep us healthy. They let researchers explore the tiny worlds inside our bodies and see how everything connects and works together. So, next time you wonder how your body functions, remember that there is a scientist out there playing excited games to find the real answer. Now, I will end today's lecture by speaking about all the things we have learned so far. So we have learned what are the cellular networks, we have learned what are biological and metabolic networks, we have learned some of the application part of it such as enzyme kinetics and dynamic modelings. I congratulate you all for opting out this particular course. Now this you might have seen, there's nothing new we are learning, we are just learning in a new prospect. The aim of this course is to see biology not as what we have learned so far, but to incorporate some of the mathematical concepts, some of the fundamental computational concept to involve in this entire biological setup and teach you a newer way to understand these phenomena. We will be learning a lot of exciting things in future classes. We will also learn some of the very basic coding, some of the very basic mathematics that will help you to learn all these biological concepts in a much simpler and a newer way. We will also have a particular session where we will teach you how to perform a simple coding either with the help of MATLAB or with the Python. So stay focused and stay tuned to this particular course, and we will be happy to receive your feedback on this particular course. Happy learning. Thank you very much.